So for today, we are going to talk about the action potential. So last class, we covered all about neurons and basic functions of cells, okay? Now we're going to extend this information to chapter 2 to learn more about the action potential and what happens when the cell actually fires, as we were talking about in the last material. Is everybody okay with the lighting? Should I turn one down? I don't want to turn them both down because then they can't see me at home. Is one down fine? That'll be a little bit. Okay, good. Okay, so everybody at home, can you see the slideshow fine? Can you hear me okay? Give me a thumbs up. Thank you, guys. Okay. So let's talk about the action potential. There is a lot of chemistry in this. There's a lot of biology in this, but I'm going to try to do it in a simple way so that everybody's on board who doesn't, maybe not necessarily have biology. I'm going to try to do this as best I can. If you have questions, you can always ask them. But here we go. All right. So let's first talk a little bit about chemistry and how it works with the cells in the brain. Okay, so when we look at how the cell communicates, we have to first understand how the cell is made up in terms of electrical components, okay? And then that will then help us understand how the action potential or cell firing works electrically speaking. So we're going to talk about two kinds of movements, okay? One is called electrostatic pressure, okay, which we refer to as electrical gradient, and one is diffusion through concentration gradient, okay? So we're going to talk about how the cells, both inside and outside the cell walls, are made up of different electrical atoms, and how the movement of those atoms and of the liquid determines of what state that cell is in. Okay, so it's accomplished through two processes: electrical gradient and concentration gradient. Okay, so electrical gradient is the same concept as using magnets. So has anybody played with magnets as a kid, or maybe even if you have access to a magnet, you're going to play with it, right? Okay, you take opposite ends, and what do you do when you put them together? If you have opposite polarized magnets, what do they do when you bring them close together? They're attracted, right? They'll go, okay, and they'll hit together. What happens if you have two different polarities and you put them together? They push each other away. They repel each other, right? So when we talk about electrical gradients, this is the same process. There are atoms that are positively charged and atoms that are negatively charged. And those with opposite charges, wait, did I say that backwards? Those have the same, so if you have, no, I said it right? Did I say that right about opposite and repel? Okay. Now I got myself questioning, okay? So the same idea happens with electrical gradient, okay? Those particles with opposite charges, so if you have a positively charged atom and a negatively charged atom, they're going to attract each other, okay? And they're going to want to get, in the same area, okay? Those atomic particles that are negatively charged, for example, they're going to repel each other. If you have other atoms that are positively charged, it's the same charge, they're going to repel each other, okay? And so if you have a liquid, okay, and you have electrically charged molecules in that liquid, you're going to see that all the ones that are the same charge are going to repel each other and move away from each other, versus if you have oppositely charged, they're going to bunch together, okay? Kind of like when you put salt mixture into water, you're going to see it disperse differently based off of the electrical atoms within the makeup of the salt in comparison to the water, okay? And it distributes differently throughout the water. And the idea is to be balanced, right? So the whole thing will eventually balance out. When we talk about concentration gradients, okay, so this is a different concept. Concentration gradient is when you see higher concentrations of something moving throughout a liquid to an area of lower concentration. What happens if you take a cup of water and you drop a drop of food coloring in it? What's going to happen to the food coloring? It's going to disperse out, and eventually, what is that cup of water going to look like? It's going to look like the color that you dropped in. So let's say you have a drop of blue food coloring, okay? When you see it hit the water, it's going to be super concentrated and very blue, right? Now, you're going to see it start to disperse throughout the water and spread itself out to areas that don't have food coloring, so that eventually the whole thing will be a lighter shade because it's all dispersed completely into that liquid area. Does that make sense? So your brain and the cells and the molecules in the cells all work with both of these processes to stay regulated, okay? And we see that these processes jump in when we start to see changes in the concentrations of the atoms and other molecules in the brain, okay? Everybody good with me so far? And understand the difference between electrical gradient and concentration gradient. Does everybody understand that? Great, okay? Now, in the brain, you have, which this menu would go away. If you guys follow along with the slides, you should be able to see the top menu. Okay. Is everybody good? Okay. So in the brain, we have fluid, okay? We have extracellular fluid that's outside of the cell, and we have intracellular fluid that's inside of the cell, okay? Extra being out, intra being in, okay? These concentrations of fluid, okay, contain different ions. Some of the most, the biggest concentrations are sodium or Na plus, right? Okay, so I'm going to probably refer to almost all of these by their actual chemical name, symbol. So just make sure that, you know, if you keep forgetting, just write it on the side, okay, of your notes. Notice that sodium has what charge? Positive, okay? Potassium in the brain has what charge? Positive. And chloride in the brain has what charge? Negative. So it's important to understand that these different molecules have different charges. Okay, that's going to play into importance a little bit later as we start to talk about the actual charges. Also, these molecules are different sizes, and we're going to get to that in a few minutes too, and why that's important. So these ions, sodium, potassium, and chloride, and other ones like large protein molecules, move in and around the cell according to those principles that we just talked about: electrical gradients and concentration gradient. Okay, they move according to diffusion, and we're going to talk about specifically how it does that in and outside of the cell. When you see movements of these ions across the whole cell membrane, okay, we're going to see the action potential. And the action potential is the firing of the cell. So when I say that a neuron is firing, I am saying that it is displaying an action potential. Okay? So those are two terms I'm going to use interchangeably. So I might say when you see the action potential, and I might say when the cell fires. They mean the same thing. Okay? Did everybody know that? Or did most people familiar with that? Kind of, sort of, yeah, maybe? Okay. I'm just trying to judge how much you guys are familiar. Okay? So, any questions so far? I'm trying to go through this kind of slowly so that everybody can have the same sort of foundations. Everybody good? Okay. Am I going too slow? Okay, good. I'm getting some head shakes. It's like, nah, just go slower. Okay. Now, remember probably back when we learned science and maybe, I don't know, 6th or 7th grade, we talked, maybe even later, I don't know. I have zero capacity of understanding science now in relation to grades. You guys remember we talked the thing, a resting potential, okay? A resting potential of something, okay? It's just sitting there waiting to be used, okay? So in the membrane of a cell, we have something called a resting potential, and it sits at about a -70 millivolts. So the resting state of the cell is actually negative. It's a negative charge, okay? So why -70, okay? If you look at different books of biopsychology and physiological psychology and all the other names that you can call it, you may see slight variations of -70. -70 is the number that we've kind of settled on for teaching purposes because of experiments done on different neurons. So if you go into this field, you might find that different neurons have different resting potentials, and it might vary. You might see some say -50, -60. They all sit at a negative level, but we're going to settle with -70. So if I ask you, like on a test, what is the resting potential of a cell? What's the answer? -70 millivolts. Okay, -70 millivolts. So how we've come to determine this particular number is through a series of experiments. And we've been able to identify this by using a recording electrode in a giant squid cell. Okay? And I'll go over how that's been done. How is it that resting potential is maintained? First of all, diffusion, electrostatic pressure, okay? So electrical gradient, concentration gradient, all factors in there. And the fact that there is leakage of certain ions across the cell membrane to keep it at a -70, okay? So there's more negative ions within the cell, okay, for it to sit at a negative resting potential. And we're going to go into what those are and how they move and how that changes the nature of the cell, okay? So we good so far? Yes. Why does the number vary? It depends on the cell. It depends on where it is in the brain. And it depends on the species, because everything that we really are basing the teaching stuff off is off a giant squid cell. So there's a little bit of a variation based on species, but generally speaking, it sits at a pretty, a negative level. And also, everything that we teach in neuroscience is like based on this study that we did 50 years ago, and it's been replicated, and we're pretty confident of it. But we're always learning new things about the brain. So you guys somebody in here might be like super into neuroscience and go into grad school and learn, well, I was taught this way, but there's a lot of exceptions to the rule. And it's just based off of what we're gathering and learning about cells in the brain. Okay, good question. Any other questions? Everybody good so far? We're going at a good pace? Okay. This stuff can be kind of heavy, so I want to kind of walk us through, okay? Because we're building up our knowledge, and then I'm going to, bam, hit you with action potential, okay? All right, so let's look at this real quick. So this is the cell membrane. Okay, so this is the cell membrane. Okay, this is what makes up the outer portion of the cell. So at the outside of the membrane, this is the actual outside of the cell. Okay, so this is all that fluid that's sitting outside of and in between each cell. Okay, here is the barrier, and then here we have the inside of the cell. So this would be all the intracellular fluid and components within the cell. So we're basically looking at inside the cell, and here's the cell here, outside the cell, and that's the outside of the membrane. Okay, everybody good so far? Now, outside of the cell, what you're going to see is there's a high concentration of chloride and sodium and a relatively low concentration of potassium. Okay? And notice that the sizes of the molecules here, or the size of the box, represent like the concentration of them. Okay? So you have a lot of chloride and a lot of sodium, and that in high concentration is pressing on the outside of the cell. Okay? So using electrostatic pressure and diffusion, it is maintaining these ions in the extracellular fluid. Low concentration of potassium. Notice that outside of the cell, it is more positive, right? Everybody see that? Outside the cell is more positive because you have a high concentration of sodium and you have a low concentration of potassium, but that's going to outweigh how much chloride is there. So it's going to maintain a positive charge outside of the cell. Does everybody see that so far? Yeah. Now, inside of the cell, we have a large concentration of we call them large proteins, which is a negative charge A. And then we have a large concentration of potassium, which is a positive charge. A small concentration of chloride, small concentration of sodium. Here, the large proteins and the amount of chloride, okay, also lead to it being a more negative charge inside, sitting at that negative 70 millivolts. Okay, and again, the forces of electrostatic pressure and diffusion are keeping those molecules in place. Large proteins cannot get through the cell membrane because the molecular size is too big. They cannot freely go back and forth. Okay? All right. And we're going to talk about pumps and how things move through here in a minute. Okay? Now, inside of the cell is mostly potassium. Outside of the cell is mostly sodium and chloride. Now, what is sodium and chloride? Salt. Okay. This is kind of gross. But if you were to lick the solution outside of the cell, what would it taste like? Kind of salty, right? Okay. So if this helps you remember that all the fluid outside of the cell is made up of sodium and chloride, and that together gives us salt, okay? And that tastes salty, okay? That if you were to lick the outside of a cell, it would taste kind of salty. I don't suggest it, but if you were, I'd give you the opportunity. Okay. So, concentration gradient allows for these ions to stay in place, potassium being outside of the cell, inside the cell, and sodium chloride outside of the cell. Electrostatic pressure explains why the inside of the cell sits at this negative 70 millivolts, because potassium and sodium want to stay inside of the cell, okay? And chloride, which is negatively charged, wants to get out of the cell and away from all this potassium and sodium because it is oppositely charged. Does everybody understand so far? So these two processes are explaining why the inside of the cell sits at a negative 70, okay? because the chloride is continuously trying to escape, okay? And the concentration gradient is maintaining the cell voltage inside and outside of the cell as well. Now, go ahead. Okay. Okay, so the question is off topic, but I'll attempt to answer it. The question is if you were to lick the outside of a brain, would it be considered cannibalism? And very odd question, but I'll take a stab at it, no pun intended. I'm pretty positive that cannibalism by definition is the consumption of human flesh. So I think if you lick it, you're good, and you're not considered a cannibal unless you're somehow getting some of that ingesting in. Very odd answer. First time I've had that question. Now, why is the brain exposed? I don't know. And perhaps the fact that you exposed it might start to teeter you over to the cannibalism realm. I don't know. But I believe it's actual consumption. So, very interesting question. Did I answer your question? Okay, great. I do not advise any kind of licking or consuming of human flesh. Yeah. Just my disclaimer, so everybody's aware. Okay. So finally, there has to be a balance of concentration gradient and electrostatic pressure in order to keep it all balanced and at a homeostatic rate. Okay. Now, let's talk about how we maintain this status. Okay. So at the top it says sodium potassium transporters. So the sodium potassium transporters help to keep the cell at its resting membrane potential of -70 millivolts, okay? Because it's constantly pushing sodium out of the cell and pushing potassium back in. Now remember, these are different concentrations of the two, okay? And how we're able to maintain the balance. And so if you look at the figure, okay, here, number one, you see that an ATP is adenosine transport proteins, which we won't really get into right now. We'll get into this later. But ATP is what we use for energy in the cell, and it requires, this process of transport requires energy. So this is why we're going to see ATP being included in this process, because without it, it can't function, okay? The cell can't work. So what you're going to see this process working here, we have step one, 3 sodium ions and an ATP being transported through this particular pump, okay? So the ATP is going to bind to allow it to actually give it energy to do this, okay? Step number 2, okay, we're going to see the ATP change shape to allow for it to process, to give it the energy that it needs, to send these 3 potassium, these 3 sodium ions out of the cell. Okay, remember, this is inside of the cell. We're throwing these bad boys out. Okay, so 3 sodiums get pumped out with the use of ATP. Okay, now you're going to see that the shape change of the transporter pump, okay, is allowing the sodium to push out of the cell, but also enables the potassium to bind, okay? So the 3 sodium get booted out, and then the 2 potassium is allowed to bind, change shape again, and push that into the cell. So for every 3 sodium molecules that the cell pushes outside, it brings in 2 potassium molecules. Now, what does that do to the charge? If we're pushing out 3 positively charged molecules and only accepting 2 positively charged molecules in, what is this doing to the cell inside the cell, charge-wise. It's keeping it negative because it's dumping out not all the positive, but it's dumping out more positive than it's taking in. Does that make sense? Everybody got that? Okay. So these very specific sodium potassium transporters are allowing 3 sodiums to throw out with the use of ATP, changes the shape of the transporter, pump, and allows 2 sodium to bind. Keep saying sodium, 2 potassium to bind. 3 sodium go out, 2 potassium come. Okay, and allows it to come inside the cell. I know I keep switching it, but you guys see it on the thing and have it right in your notes. Yes. Excellent. Another cannibalism question here.
How do we know that it's 3?
Because we can actually witness this, and you can measure the concentrations, and you can measure how much is going in and out. So we can actually take a sort of a snapshot of this at different levels and be able to tag those ions in those proteins. Okay. All right. Any other questions? Are we good? Now, this process is going to keep repeating and cycling through, maintaining that resting potential of the cell. Okay. Now, let's talk about ion channels. Okay. So, there are proteins that are embedded in the cell membrane wall that can allow certain ions to leave and enter in. And remember that the whole point of this is to keep cells at a resting potential of -70 millivolts until it is ready for the action potential to send the electrical signal through. The first step of the action potential is the opening of those sodium channels to allow for a change in charge of the inside of the cell. Now, some neurotransmitter receptors, remember these are chemical receptors, actually contain allow for quicker function and faster distribution of electrical signal, and not necessarily the signal is faster, the reaction is faster. Okay, so we're going to talk about 2 voltage gated and chemical or ligand gated types of receptors. Okay, all right, so we good so far? This is a really nice visual here. This is an ion channel that's closed, not accepting any ions in. This one right here is open, okay? And that's allowing for the change in molecular distribution. So, in order for us to make conclusions about what an action potential is and how it works, okay, I'm going to just very briefly describe the process of how we look at this and how it's studied in sort of bench science. So what do you need to learn about the neuron? Obviously, you need to start with a cell. You need a neuron. You need some way to measure electrical current in the neuron to be able to actually test this. And you need an ionic solution to store this in so that you can basically replicate what you're seeing in the brain. So this particular experiment is done with a squid giant axon, not a giant squid's axon. A squid's giant axon, the way that it's worded is actually very particular. This axon is large enough that you don't need a as highly powered microscope to use. This is why they use specifically the giant axon. And then you need the ability to be able to record the process, and you also need stimulating electrodes in order to, again, follow the process of the action potential. So this is what the setup looks like. It's actually really interesting to see this. So you're seeing that we have a petri dish with the giant squid axon here, and then we have the voltmeter so that you can actually test to see what voltage it is. We have the wire electrode that's placed in this saltwater solution, and then we're able to conduct the electricity. This is actually put into the axon itself. Then we have the stimulator, and then we have the ability to record it. This here is basically where we record the electrical impulse, okay? And then we're able to determine at the resting state, and then we're able to determine at action potential what the voltage change is, what it looks like across the axis, okay? So that's how we would do this in a lab. Yes. So there have been, you can take neurons out of the brain and you can observe them. They're so much smaller that it's hard to get a good idea of what this looks like. A lot of what we do with this, we call translational research, is we make assumptions. We say, well, the structure is the same or identical. The process appears to be identical when observing it in other fashions. So that's kind of like taking what we know and building on it based off of these kinds of experiments. If you take sensation and perception with me, you'll see that we do the same sort of thing with animal work and looking at things like the visual system, hearing, things like that. These experiments are done with a brain as close as we can get to a human brain, which is usually non-human primates, and looking at the structures and the makeup and saying, well, these are basically identical. So you take the information that you learn there and you apply it to the human brain. So we're taking what we learn on a larger scale and applying it to the same cell structure as a human and saying, this is how it works. Does that make sense? Any other questions? You good? All right. So, what did we learn from doing these experiments on a squid's giant axon? Okay. So we've learned that the resting potential, okay, is a different charge when the neuron is at rest versus when it's active. Okay. So we're settling on that negative 70 millivolts being the resting state, and then that the inside of the cell is charged negatively in comparison to the outside of the cell, which is a positive charge. Any stimulus that elicits an action potential, which we call a sufficient stimulus that crosses that threshold, is caused by the movement of those positively charged ions, the potassium and the sodium. We know that the axon is what carries the electrical impulse to the end of the neuron. And we know that the change in the membrane's potential in charge happens as it moves down the axon, okay? So you actually see it change from a negative 70 millivolts to a positive charge as it moves that electrical impulse down the axis, okay? I have a YouTube video on here. It's not a cartoon, but it's like a computer graphic of what an electrical impulse probably looks like in a human brain. So that's just a neat way to kind of visualize how we do that. Has anybody been able to access the self-service human anatomy app yet? Is it on there? Excellent. So you guys now have access to the human anatomy app. It's called Human Anatomy Atlas. It should be able to be installed from self-service. If you don't see it, let me know, but you should be able to see it because if you're enrolled in the class. We won't be messing with that yet because we can't look at molecular stuff on it. We can only look at kind of big brain stuff. So we'll be using that in class probably next week or the week after. Okay. Any questions so far? Are we good? All right. Now, let's look at what the action potential looks like on a graph. Okay. So when you change the inside of a cell, a negative charge, to a more positive charge, okay, you're changing, when you change the charge, you're basically causing it to engage in an electrical stimulation that follows all the way down the axis of the cell in the next cell, stimulate the next cell. And if it's a strong enough signal to the next cell, it will then potentially cause the next one to fire as well. Depends on where it's attached to the cell, the next cell, and what the other cell is doing. Okay, so we're going to talk about that here in a minute. Here's what it looks like on a graph. So this is the threshold. Okay, so this is right here. This is the threshold. This right here is the resting state. So if you look at where the cursor is pointed here, okay, everybody's state of the cell is sitting at a negative 70 millivolts. Okay, so this cell is just sitting there minding its own business. Nothing's happening. It's just resting. Okay. Now another cell's connections, possibly connected onto the cell's dendrites or cell body, okay, is sending a signal to the cell saying, hey, wake up. Okay. So what we're seeing is, hey, wake up. So we got a hit, that did nothing. You see that? Okay, we got hit number 1, that did nothing. Hit number 2 did nothing. Hit number 3 did nothing. Still maintaining its resting state. So each time this cell gets hit, its charge changes a little. So each time it gets hit, the charge are starting to make this cell a little bit more positive. So hit number 1 changes it from a negative 70 to maybe like a negative 65. Okay, but that's not enough for the cell to respond. Right? Then another hit, another stimulation, more sodium and potassium come in, okay? And for it to hit, so the threshold in this case, and again, this is something that you might see the number change a little bit based on the book. In this case, the negative 60 is the threshold of excitation. So when it hits, when the charge changes from negative 70, and in this graph, in this case, to negative 60, it starts the process of the action potential. Once the process starts, once the threshold is hit, it's an all or none thing. Everything starts to work. It doesn't need any more stimulation. It doesn't need any more signaling. It doesn't need any more messages. The cell takes over from this point, okay? Now we're starting to see ions flood the cell, positively charged ions flood the cell. So if the cell was at a negative 70, minding its own business, it got enough stimulation to drop it to a negative 60, okay, or to increase to negative 60. Okay. What happens to the charge if positively charged ions rush the cell? Does the cell become more positive or more negative? It becomes more positive. So if it's sitting at a negative resting state of negative 70, it gets stimulation to set it at a negative 60, it's becoming more positively charged. Now it hits the threshold and all the sodium and potassium rush in. Now it's going to change the charge of the inside of the cell to be more positive. In fact, it happens so fast and there's so many ions that rush in positively charged. It passes 0. So it goes from the resting state of -70 to the threshold of -60, hits the switch. Now it's going to rush in to hit a +40. So it's going to get so positively charged that it's actually going to cause the inside to now be positive. Does everybody see that? So starts at -70, and then the peak charge 40. Okay. So if I were to ask you, what's the peak positive charge that the cell membrane gets to, okay, the membrane gets to, what is your answer? A what? You can hit it at home too if you want to answer as well. +40. Very good. +40. So once it hits that peak positive charge, okay, once it hits the peak positive charge, it will then hit its max and start pushing out those positive ions, okay? So it's going to push out the positive ions. What happens to the charge of the cell if it's pushing out all the positive ions? What does the cell now become? More negative. Exactly. So now the cell is becoming more negative. Okay. And then it hits the resting state again at -70. But this process, remember we all talked about regulation, homeostatic regulation? It actually overshoots in this process of hyperpolarization and actually dips below even more than -70. It hits about -80 or so, and it's got to kind of regulate itself back to -70. Okay. And during this process of hyperpolarization, okay, it does not have the ability to fire again. It's got to regulate itself. Okay. And we'll talk about this in just a second. Does everybody see that? As the cell gets the stimulation, it's going to change from a -70, hit the threshold to -60 or so. That's going to start the process. It's going to change its charge to a +40, and then it's going to dip down all the way below -70, and then it's going to be upright again. And now at this point, it's ready to go to fire again. And this whole thing takes about 3 milliseconds. Okay, and it fires over and over and over again. Yes. Yeah, I have another graph now, and so that'll explain that. Yeah. Okay. Any questions so far? Are we good? I should have another graph that actually has 3 terms on it. So if I don't, I'm going to put that in there because it's really easy to tell the difference between the 2. All right. So here is the steps. Here are the steps at each point. Okay. Here. All right. So we're at -70. The threshold of -60 is hit. At this point, the sodium channels open, and then sodium starts to flood the cell. And then at step 2, the potassium channels are going to flood the cell. So first we have the sodium channels opening. That's going to start getting it to a more positive charge. Right after that, the potassium is going to open up, continuing to make the inside of the cell more positive. And then here, the peak is met. Now the sodium channels are refractory. They can't do anything right now. They're kind of stuck in this refractory state. So no more sodium enters the cell. So then that means that the cell can't become more positive because it's not accepting in any more positively charged ions. Then what we see is that potassium, so that peak is hit, potassium will start leaving the cell. And we have that the potential starts to go to resting levels. This step here, the potassium channels are closing, sodium channels are resetting, but in the process of those sodium channels resetting, it's going to dip a bit below, and then those extra potassium ions, are going to then diffuse and get it back to the negative 70 resting state. Okay? Everybody good so far? And here again, here are the different steps here. This is step 1 with the channels opening. Here's the refractive period where it's closed. And then the sodium channels are then reset. Okay? Allows for us to do it over and over and over again until we die. Okay. Any questions about that so far? Good? See, we have repolarization, hyperpolarization, and depolarization. And I thought I had a table on here that showed all three. I'm gonna have to go back and edit it. Okay. When it comes to refractory periods, okay, there are 2 types of refractory periods that occur after an action potential happens. So we have an absolute refractory period where the cell is essentially halted for just about a half a millisecond, where it can't do anything else. Okay. How many of you have ever flushed a toilet and then realized that you probably need to flush it again for whatever reason? Okay. And you tried to do the little flusher and it just went bloop, bloop, bloop to you. Anybody ever experienced that? Okay. What that toilet is doing is experiencing an absolute refractory period. What has to happen to the toilet for it to be able to flush again? It has to fill back up with water in the back, right? So in the process of it filling back with water, that toilet's going to do nothing, okay? You're just going to have to sit there and watch, right? And then once the toilet's ready and filled back up with water, then when you push the when you what is that called? Flusher, handle, right? Once you do the handle of the toilet, it's going to then flush again. Same process with the neuron. It goes through this absolute refractory period where it cannot fire again. It just physically can't. Now, why do you think we should have an absolute refractory period for our brain cells? Why is this important? It needs time to rest. Absolutely. What happens if a brain cell can't rest? Okay, what if it can't go back to homeostasis? What happens to the cell? I'm hearing some mumbles. It burns out. Essentially, it gets fried. What happens to fried cells? They die. Right, exactly. So when you have cells that are firing too often and not experiencing this absolute refractory period, essentially what you're having is you're burning your cells out. And that's not a good thing. That's not a healthy cell. Okay. You can actually see this happen with conditions like epilepsy. Okay. So epilepsy is when you have excessive firing of neurons. Okay. That's not good for your brain. It's not good for your brain to have this excessive firing in areas that aren't supposed to be firing right now. Okay. And that is not that's not going to be conducive to a healthy set of cells for your brain. Now, we also have a relative period, which we call the relative refractory period, where it's unlikely that the cell can then fire again. It can happen, but there is a period of time where it is in a resting period where it normally does not fire. Okay. And what drives these are the opening and closing of the sodium channels. Okay, so in cases where we have closed off channels, we can't have that rush of positive ions, and therefore it will not allow for it to change in charge to allow for an action potential to occur. Okay. Any questions about that? Yes. That's a good question. So what cells have the absolute versus the relative refractory period? I want to say, I don't know the answer off the top of my head. I don't know if it's always cell specific. I think it's more sometimes location specific, and I think it's based off of some motor versus reflector cells. I think we might cover this a little bit later, but I can't think off the top of my head one having one over the other. It's more of like there's a period of time right before the absolute period where there's a refractory, and then a period after that's, you know what I'm saying? So that's probably a better explanation of the two together than cells having separate things. Yeah. Any other questions? Yeah. What causes the change in the status of the positive versus negative? Okay. So the whole thing starts with a stimulation from another cell. So, and we're going to get to that. So you'll see. So the exchange of information from cell to cell is chemical. That chemical message could say fire or don't fire. When enough message is received, that stimulation will signal the cell to have an electrical impulse, and that's where this all occurs. And we're going to talk specifically about what is enough and where it is in order to get this process started. Okay. All right. Any other questions about refractory periods or the process in of itself? Are we all good so far? Okay. Now we're good. Give me a thumbs up if you're following along. Yay! Great. Thank you, guys. All right. Now, very important feature of the action potential is something called the all or none law. Once the action potential begins, once it is triggered, it goes down the length of the axon. It either happens or it doesn't. We don't have a partial action potential. We don't have a graded action potential. We don't have a sort of kind of action potential. We either have the message is sent or the message is not sent. Okay? One or the other, all or none. It either happens or it doesn't happen. Now, we do have something called the rate law, and that's about intensity of the stimulus, and that can change the rate and variation of firing. So the rate law is not does it fire or not necessarily, or how fast it fires. The rate law is lots of firings back to back, or slow firings. It's not like how strong each one is, it's the rate and frequency of the firing. That's the rate law. Okay? So variations in intensity, how fast and how quickly are they from back to back, as well as variation in the rate of the axon itself in terms of firing. Okay? So here we see, okay, a shorter period versus a longer period wait of actual firing. Does that make sense? And that is taken through the giant axon of the script. Okay? Any questions so far? Are we good? All right. Now, we've talked about this a little bit before, but this is where we're going to explain how this works with the myelin sheath. Action potentials are regenerated down the length of the axon. So as it hops from node to node, we're seeing those ions move in the membrane of the axon continuously to allow for that electrical impulse to continue down the length of the axon. So what you're seeing here is that the sodium and potassium channels, okay, open along the length of the axon, okay? They open, they open, they open, they open. here, 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 open here, 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 open here, here, here. And at each section of the exposed axon, okay, we're seeing this process repeat. And we call this a regeneration because we're essentially recreating the action potential at each open spot. Because the myelination occurs, the myelinated fatty tissue here is going to change the conduction, okay? And it can't do it in that particular spot. So it's got to regenerate and jump nodes at every point of the nodes of Ranvier. So here, these nodes of Ranvier allow for the electrical impulse to continue to regenerate until it hits the end of the axis, okay? So the myelination or the myelin sheaths are going to increase the speed of conduction and allow it to go faster from node to node, okay, through saltatory conduction. So at this point, there are fewer sodium and potassium channels that have to open at each node, allowing for faster conduction. If we had no myelin sheath, imagine this. We have no myelin sheath. That would mean that every sodium and potassium pump and channel along the entire length, its job would have to be open, 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 open. That would take a long time, and it's not smooth. And if we have a disruption, that's going to cause a problem for that electrical impulse to go down the length of the axis. However, if we only have sections of sodium and potassium channels that have to open at any given time, it's going to allow for faster processing of those ion movements to allow for faster conduction down the length of the axis. Because now we get open, 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 open versus open, 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 open. You see how that changes? How fast that message can jump from node to node? If we're like open, 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 open versus open, 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 open. So the myelin sheath are giving it the opportunity to cover the cell in such a way that speeds that electrical impulse up from node to node. Does that make sense? Is everybody seeing that? Any questions at home? So everybody see how the myelin sheath works to allow for less surface area of the nodes of Ranvier to require those sodium potassium channels to open along the way. So to summarize all of this together, okay, the activation of a neuron through the signal from another cell and enough stimulation occurs to cause an action potential to happen. The action potential will then propagate down the length of the axon, jumping from node to node of the nodes of Ranvier to the end of the cell. What happens now when it gets to the end? So the cell has got this electrical impulse that runs throughout it. Now it's going to hit the end of the cell and then release the chemicals to send a message to the next cell, whether or not to fire, and what kind of chemicals for it to release if it does fire. Okay? So it's a complex message of what the next cell's job needs to do. And the kind of cell and the location of the cell is going to give us information as to what kind of chemicals that cell is producing and synthesizing and what message is getting sent from point to point. Okay? So it's going to depend on what kind of cell it is. It's going to depend on what kind of chemicals it's synthesizing to determine what that message is going to be and in what quantity. Okay? So if we look at this right here, this is a sending cell. This is the terminal button. Here we have the microtubules that's coming from the axon. And then we have different ways that the chemicals can be transported and synthesized, whether it's the cell body, whether they get transported through the axon, or whether they're synthesized and housed here in the terminal button. This is the receiving end. This is the dendrite or cell body here, the postsynaptic area, where it's going to then receive the chemicals from that action potential message. Does everybody get that? Does that seem clear? Then we're going to go over this process and how cells get this information and what they do with it. Okay? Everybody good so far? We'll do one last slide and then we'll end for today and I'll open for questions. Okay. So the very crux of this, why do we care about the action potential and then sending a signal? The cell in of itself has sent the action potential. Now what does it do? The next cell has to determine if it's going to also fire an action potential, but it takes the information from the previous cell to determine that. Okay? So in order to receive information in chemicals, it has to have the ability to have receptors to accept the kinds of molecular shapes that the cell is trying to give to it. So here we have the terminal button of one cell, the dendrite or cell body of the receiving cell. Okay, so the message has been sent and this electrical impulse has activated the cell. The action potential occurred. Now we have these chemicals to relay another message. So this right here is the presynaptic membrane. Okay, and it contains vesicles or sacs that contain chemicals known as neurotransmitters. The neurotransmitters are things like dopamine, serotonin, acetylcholine, choline. These are all different kinds of chemicals. Okay, and then here we have the synaptic cleft or synaptic gap, which is essentially a very, very tiny open area. It's not physically attached to the cell. It's an open area, physical space between neurons where the chemicals then get sort of exchanged. And then finally, we have the postsynaptic receiving end, and this has receptors that are designed to take in very specific chemical structures. So if you have a cell that's giving you dopamine, you have to have a dopaminergic receptor to accept that chemical message. It's like a lock and key. So think of the chemical like dopamine as the key, and the lock would be the receptors that are on the cell. They got to lock into place in order for it to take that information. Okay? So next time we will talk about specifically the vesicles and how the chemicals are synthesized within the cell, where they're synthesized, and all that. Does anybody have any questions about the material? Everybody good? Okay, at home, does anybody have any questions about the material? All right, I'm going to go ahead and end the recording.